Hello everyone, today we are going to see about vitamin A, and this is one of the fat soluble vitamin, and in this session we are going to see about what are all the vitamins of vitamin A, what are the sources, functions, deficiency, RDA, and the functions, metabolism also. So coming to the vitamin A, it is a fat soluble vitamin and it is mainly essential for eye vision, growth and immune function. And it is found as true vitamin A as retinol in animals and provitamin A carotenoids, that is beta carotene, in plant source, which is in animal foods it is present as retinol, and in plant sources it is present as beta carotene. And coming to the Vitamers, it is having 4 basic Vitamers. First one is retinol, which contains the alcohol group, and retinal, which contains aldehyde group. Retinoic acid, it contains carboxylic acid group, and beta carotene, it is the provitamin A. So retinol, it is the primary alcohol along with beta ionic ring, and it is mainly used as storage and transport. And coming to the retinal, it is aldehyde and it is the oxidation of retinol group, and it is most important for vision. Retinoic acid, it is mainly important for the gene regulation and growth. Beta carotene, it is a plant precursor which is converted into retinal as beta carotene. Coming to the chemistry, retinol contains beta ionic ring and isoprenoid chain. Beta carotene is cleaved into 2 molecules of retinal in the intestine, and retinal cannot be reversed back to retinol, but retinoic acid cannot revert back. And this is the structure where you could see the final functional group retinol CH2OH, aldehyde CHO, aldehyde functional group, retinoic acid COOH. Beta carotene have 2 beta ionic ring in one side and another side. And coming to the vitamin A and vision, and it is achieved in the form of Wald's visual cycle. In our eye, retina is present, right? So this retina contains rhodopsin as pigment molecule, and it is present in rods of the eye. And this rhodopsin contains 11 cis retinal and opsin. These two forms contains the rhodopsin. So when light rays hits our eye, 11 cis retinal isomerizes to all trans retinal. That is, isomerizes, it just changed the structural shift from cis to trans. So cis retinal is the same side and trans retinal is the opposite side. And this triggers conformational change, and just nerve impulse happens and sends signals to the brain, and it instructs our eye to see the light. So all trans retinal converted back into 11 cis retinol and combines again with the opsin pigment. So wherever the incomplete conversion is there, it should be sent to liver as retinol and further converted into 11 cis retinal. So this is the rhodopsin cycle or Wald's visual cycle. And Wald's visual cycle, you could see the 11th rhodopsin, which is consists of 11 cis retinal and opsin. When light photons hit the rhodopsin, through the nerve impulse, it gets converted into all trans retinal. In that, all trans retinal converted into 11 cis retinal. Through alcohol dehydrogenase enzyme, all trans retinal converted into 11 cis retinal, and 11 cis retinol converted back into 11 cis retinal, and it again combines with the opsin, and the cycle gets continued for further seeing the light reaction. And this is the only cycle which has happened in liver, and which has happened in our eye visual cycle, and without any conversion it should be sent to the liver for further conversion. So you could see another image also where light energy hits eye, it sends signals to the brain, and the retinal and opsin separate and just recycled back and further joins with the rhodopsin and further joins with 11 cis retinal and opsin, and the cycle gets continuous for seeing our vision.